Hello, welcome to my capstone project on regenerating Pacific Northwest salmon habitat through riparian restoration. My name is Felicity Strzelec, and I did this project in collaboration with Whale Scout, of which my site supervisor was Winnie Negebauer, and my faculty advisor was Jason Toft. So let me give you some background on the issue of Pacific Northwest salmon habitat. So first of all, humans are reducing river riparian zones through urbanization and developmental activities. And for reference, the riparian zone is the area between the river and land. And also, riparian vegetation restoration efforts are currently taking place to revitalize salmon habitat. However, monitoring how the riparian vegetation is maturing in salmon habitats is important for understanding the benefits of riparian restoration to salmon. Therefore, riparian restoration analysis, which is my capstone project, can be used to demonstrate the effects of riparian regeneration on salmon habitat. For this project, my main research question was, how does riparian vegetation affect the habitat of Pacific Northwest salmon? And I researched this through my internship and methods of riparian restoration on Bear Creek, which is located on this map in Redmond, Washington, and on the Sammamish River in Bothell, Washington. I also did a literature review on riparian ecosystems to get some scientific background on how the riparian zone functions in relation to riparian restoration. And I also completed vegetation monitoring to test riparian restoration effectiveness on salmon habitat as part of the internship deliverable. So figure 1 is showing the route of salmon in Washington. So they first start in the Puget Sound and then go through Ballard Locks in Seattle, and then through Lake Washington up through the Sammamish River. And then either they continue through the Sammamish River or some of them go through Bear Creek, and they all eventually end up in Lake Sammamish and then go on to Issaquah and further after that. So I found during this internship that as riparian vegetation increases in height, it creates more canopy cover over salmon habitat, which can be seen in figure 2 and 3, where figure 2 shows the average heights of native trees over the course of 4 years, and you can clearly see that the heights have increased over 4 years in which riparian restoration was present, therefore the canopy and cover increased. In addition, the average heights of native shrubs also increased over 4 years, as seen in figure 3, and therefore the canopy cover also increased for shrubs on the riparian zone of Bear Creek. I also found that riparian vegetation improves woody debris and litter mass in salmon habitat, which can be seen in figure 4 and 5, which I will explain soon. And for reference, woody debris is just branches and twigs on the side of rivers that helps to add nutrients to the water and also cycle nutrients through the greater ecosystem. Litter mass is the layer of organic plant material on the sides of rivers that also contributes towards nutrient cycling, and bacteria also decompose important nutrients that go into and out of the system. I also found that riparian vegetation provides a natural barrier to protect salmon habitat, and this can be seen in figure 4 and 5. So figure 4 is showing the riparian restoration that I did on the Sammamish River from July 2023 to October 2023. The left image was taken in July of 2023, and the right image was taken in October 2023. The image on the left is showing a cleared area after the invasive species of blackberries was removed, and then it shows a layer of mulch and cardboard that was added onto the cleared area to prevent the emergence of the blackberries, to help prepare for the native tree planting, which can be seen on the right image, and that's showing native trees that were planted there during October of this year, such as red alder, western red cedar, Sitka spruce, and Douglas fir. And figure 5 is showing the progression of Bear Creek from 2018 to 2023, and basically you can see here that the woody debris and litter mass has greatly increased from 2018 to 2023, while the image on the left is very sparse in vegetation and canopy cover. The image on the right is very dense in canopy cover and litter mass and woody debris, which is helpful for salmon habitability. So my key takeaways from all this, like basically synthesizing both the graphs and the images here, is that canopy cover from riparian vegetation adds shade to the riverside, which cools water for salmon. And this is important for salmon in Washington because salmon are a cold water dwelling species. Therefore, they thrive best in temperatures that are between 40 and 50 degrees Fahrenheit. I also found that woody debris increases the availability of insects for juvenile salmon. So when salmon are in their developmental stages, they feed on aquatic insects that tend to populate on the side of rivers where there's a lot of branches and twigs and leaves. So therefore, having more woody debris allows a food source for these juvenile salmons and therefore helps to enhance the greater population of salmon in Washington. Another takeaway is that litter mass from riparian vegetation provides a filtering mechanism against toxins that may seep into rivers from roads or buildings. So basically just having a more denser barrier between the vegetation between the river and the land helps prevent the seeping of pollutants into the water, which can be harmful to salmon. So the broader significance of all this is that riparian restoration can help control the emergence and spread of invasive species in salmon river habitats, such as blackberries, which is shown in Figure 4. And another broader significance aspect is that planting of native trees along salmon rivers and streams can improve salmon spawning, migrating, and rearing in developed areas, especially where there's a lot of buildings and roads. And finally, riparian vegetation monitoring and analysis enables a better understanding of how to use the riparian restoration technique to strengthen salmon populations. I would like to acknowledge and thank my site supervisor, Whitney Negebauer, and faculty advisor, Jason Toft, as well as my friends and family for helping me throughout this whole capstone experience and process. Thank you.